Hello, folks. So, Chapter 13 is all about transcription, and this is a pretty natural process that we're moving in now that you guys have been through Chapter 12, which is DNA replication. Some of the overall big processes of transcription are going to be similar to DNA replication, but the products are going to be completely different. So, with transcription, we are concentrating on creating a new messenger RNA sequence from a template DNA sequence rather than with DNA replication, we were worried about copying the template strand of DNA. The major steps of transmission or transcription are very similar to DNA replication. The process begins with initiation, where we have a transcription apparatus, which we're going to describe in just a few minutes, that is going to assemble on the promoter region of the DNA template and begins the synthesis of creating that new messenger RNA structure. The second step is elongation, where RNA polymerase, so another enzyme, but instead of it being DNA specific, it's more specific to RNA, is going to move along the DNA template and is going to keep adding RNA nucleotides to that 3' end, very similar to how DNA polymerase 3 did so in DNA replication. And then finally, the last step of transcription is termination, and this is where the building of that new messenger RNA strand is going to terminate or stop, and that process also describes the separation process from the original template DNA strand. So if we look just very briefly at initiation, in bacterial cells, it has a multi-step process. So with the initiation process that happens in bacterial cells, if we look at the top of this figure here, we have a factor, a sigma factor, that is going to associate with the core enzyme on that template DNA strand. And that core enzyme and the sigma factor together are going to form what we call a holoenzyme. And that holoenzyme in step 2 is going to bind at very specific places above the promoter sequence, specifically at 35 base pairs ahead of the promoter, and then again at 10 base pairs ahead of the promoter. And that consensus sequence in the promoter region is going to create a very closed complex, creating a lot of specificity for certain promoter regions and holoenzymes. The next step, the holoenzyme is actually going to bind the promoter region very tightly. It is going to unwind that double-stranded DNA and creating an open complex, very similar to what we see in DNA replication. And then the next step is that a nucleoside triphosphate that is complementary to the DNA at the start site is going to be the very first nucleotide in the RNA molecule. And that after that RNA molecule has been started, then in step 5 we see 2 phosphate groups are going to actually be cleaved from the nucleotide triphosphate, creating an RNA nucleotide that is going to be added to the 3' end of that newly synthesized messenger RNA molecule. And finally, in step 6, that sigma factor that originally bound in step number 1 is going to be released, and the RNA polymerase is going to move beyond the promoter region to continue to lengthen that new messenger RNA strand. So the take-home message from the initiation step in bacterial cells is that RNA transcription is going to be initiated when the core RNA polymerase binds to the promoter region, and that is going to be more efficient with the help of a sigma factor. So when we look at the eukaryotic cells transcription initiation process, we have a little bit of a few differences. And keep in mind, eukaryotic systems have much more genetic material, therefore they need to be more sophisticated and therefore are usually more complex. So, with eukaryotic cells, the initiation process in transcription, we actually have multiple core promoter sequences. And those promoter sequences are just going to create more specificity and more complexity so that specific genes have to have um, many different checkpoints so that they are not turned on sooner than they need to be. And then eukaryotic cells also have a regulatory promoter sequence that resides upstream and downstream from the promoter region. So, if we look more in depth at the transcription apparatus that is really going to come together and assemble in the initiation process, that's made up of 5 major things: RNA polymerase, transcription factors, some mediator proteins, regulatory promoters, enhancers, and silencers. And so, regulatory promoters are going to either have the ability to speed up or slow down transcription. So they really are going to regulate how fast transcription takes place. Enhancers are going to increase the rate of transcription, so they're going to also help to make the transcription process more efficient. And those enhancers can actually reside either upstream or downstream from the gene that is being promoted. And then finally, silencers are going to decrease the rate of transcription. So when we look at the overall initiation process in eukaryotic systems, there's all of these things that have to come into play. The big one is that we have to assemble this transcription apparatus. So all of those different pieces that was mentioned on the previous slide have to come together and work together to actually start the process of transcription. So if we just very briefly look at this figure here, we've got transcription factors that are going to bind to what we call the TATA box in the promoter region. The TATA box is a very specific sequence of nucleotides that are in the TATATA orientation. So that is going to signal RNA polymerase to actually come in and start transcription at that promoter region. And then the holoenzyme, when it assembles, it is going to consist of the RNA polymerase, transcription factors, as well as the mediators. And those 3 things are going to bind to the transcription factors, starting to create the foundation of this apparatus. Finally, activator proteins are going to bind to the specific sequences on the DNA template strand, and they're going to enhance transcription process. And what ends up happening with the original DNA strand is it's going to create a loop like we see down here in step number 5. And that loop really allows the surface area of the DNA to expand so the proteins can actually come in and bind to the template DNA to enhance the interaction with the apparatus. And finally, in step number 6 of this process, that those transcriptional activator proteins are going to bind to specific sequences in the promoter region and interact with basal transcription rates. So the second major step is elongation. Now that we have this very intense step number 1 where mRNA is going to be initiated, or the synthesis of mRNA is going to be initiated, we now have to elongate that mRNA strand. And some of the major substeps of elongation is that RNA polymerase is going to bind to that template DNA strand and is going to move downstream. And it's going to keep adding nucleotides, RNA nucleotides, to that 3' end of that new messenger RNA strand. This process is much, much slower than DNA synthesis, like we talked about in chapter 12. And that really has to do with all of the different signals that are happening in a cell that are either telling the enzymes involved in this process to either turn on a gene or turn off a gene. So that whole process requires much more extensive communication. Therefore, the rate of transcription is much slower than DNA synthesis. So, with the elongation process, after we initiate RNA synthesis, we don't need a primer region after that. So, the new nucleotides are going to be added to the 3' end of the RNA molecule, like we see in step number 2, where we are elongating this new messenger RNA. And during that time, DNA is going to continue to unwind at the very front of the transcription bubble. And then after DNA unwinds, we aren't creating a new DNA template, we're creating messenger RNA. After the unwinding process and the elongation process, DNA in the back is then going to start to rewind into that double helix because DNA does not like to be single-stranded. And finally, the last step in transcription is termination. And this is where the polymerase enzyme is going to move along the template to a termination site. And that's going to be a specific sequence of nucleotides. And there's 2 main types of termination. It can either happen with row-independent or row-dependent mechanisms. And these two mechanisms operate differently, but the result is the same. mRNA synthesis comes to an end. So we'll investigate row-independent or dependent a little more in depth within the lecture. But for your information, row-independent really looks for a specific consensus sequence. So it's looking for a sequence of nucleotides that are in a repeated order. So it might be something like we see up here, where we have A's, A, G, C, 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 C. So we get nucleotides that are going to be repeated in a long chain. And that is what the independent enzyme and mechanism really looks for. On the other side, row dependent is where an actual protein will come in and terminate the synthesis. So row independent really looks for a consensus sequence. Row dependent actually is where another protein comes in and terminates the sequence. So what we end up getting is, if you think about this in terms of a whole cell component, within that nucleus, we've got our DNA template is being transcribed from DNA into mRNA, and that new mRNA strand is the green strand here. And that DNA is still very active, so it's unwinding for the elongation process and then rewinding. And everything around that DNA strand, the component that's actually holding that together is RNA polymerase 3. So that's what this big yellow structure is a one big, huge protein. So some of the major differences that we see between bacterial and eukaryotic systems is that with eukaryotic transcription, there's 3 different polymerases that are involved. So because there are more polymerases, there's more activity and more specification that has to take place in the initiation step. All right, so that is chapter 13. Make sure to take your prep quiz as well, and we will see you in lecture.